we do if we want to know the level of support for a certain psychological finding? A meta-analysis is one way to get this information. A meta-analysis involves going through all of the studies that have been published on a topic, and ideally all the studies that have been run that also haven't been published, and extracting findings and combining them quantitatively and mathematically to get a sense of the direction of an effect and how big the effect is, and whether the effect is real at all. This is in contrast to a narrative review, which is the kind of review you'll often see in introductions for psychological research papers. In those kind of reviews, a researcher reads through the literature and writes a summary that explains their view of the literature and why they see their research question as important in the context of the literature. Well, what's wrong with that approach? Well, nothing is wrong with it, but it does have some important drawbacks that a meta-analysis can help to address. One drawback is that researchers are humans just like everybody else, and we're subject to biases, confirmation bias, availability heuristic, and so on. Confirmation bias is that we see what we expect to see. Availability heuristic is that we remember the things that stick out most clearly, even if those things aren't actually the most likely. So we might remember the findings of an especially flashy study, even if it turns out that that study was an anomaly and that most other studies didn't actually come to that finding. What does a meta-analysis do differently? In a meta-analysis, a researcher or a team of researchers will come up with a set of strict criteria that they will use to determine whether studies are included or excluded from this synthesis that they're creating. And that means other researchers can look at those criteria and decide whether they think those criteria are biased. If peer reviewers and other researchers seem to think this is actually a fair way to review the literature, then we can be more confident in the results of the meta-analysis. Meta-analyses also have the advantage that they usually rely on effect sizes instead of p-values. An effect size is a measure of the magnitude of a difference between groups or the magnitude of a relationship. So a correlation coefficient, Pearson correlation R equals 0.4, that's an effect size. That tells you how strong the relationship between two variables is. That's about a moderate correlation. We could find a statistically significant difference between an experimental group and a control group, but the actual effect size could be very small. Now, from a scientific perspective, it might be interesting to know that those two groups are different, but if we're thinking about designing a treatment, we don't want to be relying heavily on tiny effect sizes. That's not going to give us a lot of bang for our buck in actually trying to make a real difference for people. That's why when you see media reports of a statistically significant relationship between two variables, you want to take that with a grain of salt. In an enormous sample, it's possible that even a teeny tiny relationship, if that relationship is actually real, could show up as statistically significant. On the flip side, studies with clinical samples, for example, people who are looking for treatment for an anxiety disorder or people who are currently getting treatment, those studies often have smaller sample sizes because they're harder to run, they're more expensive, and they take longer. If a study is too small, has too few participants to be able to detect statistical significance for an effect size, we say that that study is underpowered. That doesn't mean that we want to completely throw out those studies, though. There's some valuable information in there. We just need to know what it is, and we need to take it into consideration with the other studies that have been conducted, asking a similar question and ask whether they all together come to a clear set of findings. So, does that mean you should automatically trust the results of a meta-analysis more than the results of some other kind of synthesis of the literature? Not necessarily. You should read it, and you should always read studies with a critical eye. It may be that the researchers have a very skewed set of criteria that they use to pick their studies. Or they might not have accounted for the file drawer problem, which means the fact that studies with null findings or that find smaller effects are less likely to be published. The goal is to get all of the studies that are relevant to the topic that you're interested in that have been conducted. Now that may not be possible, but you want to get as many as you can, and you don't want your findings to be skewed by whether those studies were considered publishable. Some things to consider when you're reading a meta-analysis. Do the search criteria that they used look reasonable? Are these the criteria that you would have picked to find all of the studies that have been conducted on a topic? If not, what's missing? Do you think that might have actually possibly impacted the results of the meta-analysis? If the answer is no, then keep reading. It might be that this is a difference that doesn't really matter. If the answer is yes, then you should take that into consideration when you're interpreting the findings. Something else to consider: how many studies are being included? And what's the quality of those studies? There's an old phrase, garbage in, garbage out. If you compile a lot of studies that have the same methodological flaw, you're going to have a set of results that is going to look like a finding that's real, when actually it's going to be the product of the same flaw that was in all the studies that went into the meta-analysis. A meta-analysis does not get rid of poor study design. What's a good effect size? Well, that depends. There are some rules of thumb. A Cohen's d of 0.2 is considered small, a Cohen's d of 0.4 is considered moderate, and a Cohen's d of 0.8 is considered large. But whether those differences are actually clinically significant is going to require a better understanding of what you're actually measuring and what those differences actually represent in terms of the severity of symptoms